Morning, everybody. Welcome back from the break. Everybody all well rested? Okay, whatever. All right, just the home stretch now. And this week, I'm going to ask faculty who are teaching the data science classes next spring to stop by if they can. And the first person visiting is Professor Adam Lucas, who will be teaching Statistics 88, which is a connector.
Hi, everybody. So my name is Adam Lucas. Hi, I see Nushi there. Hi. I just want to introduce myself really quickly because I'm going to be teaching Stat 88 in the spring. Could I just see a show of hands? Is anyone planning on taking that? Okay. Well, it's a great course, and I encourage some of you to consider taking it in the future. Are any of you wishing to take it, but maybe you didn't get in or you're waitlisted? Okay, so let me just say a few things about that because the course is full and the wait list is also full, but and I won't be able to manually take people off of the waiting list or expand the size of the course just because of limited resources. But if I do know that they check the waiting list at 7 a.m. and 7 p.m. and you can check. So they update the waiting list at that time, and you can check shortly after to see whether that has moved at all. And I guess once classes start. But anyway, in Stat 88, for those of you interested in taking it in the future, it's basically a connector to this course, which basically goes into some of the technical aspects of what you've been doing, in particular probability and regression and spread of data. So all those things, it's really important to have an understanding of the foundations of what you're doing. You're doing great data science now, but as you know, lots of people misuse statistical methods, and so it's important to understand where it all comes from and what assumptions there are behind the tools that you're using. So anyway, I'd encourage you all to consider taking it sometime in your career here at Berkeley, and those of you that will be taking it in the spring, I look forward to seeing you. Oh, yes, I'm sorry. Does anyone have any questions? Yes, hi. It's a 2-unit class, so it's half. It meets once a week for 2 hours. Other questions? Okay, well, thank you.
So I thought I'd turn that off just to stop this, but apparently not. Can you hear me at the back? Yes? Good? All right. So Stat 88 is dear to my heart because I developed it last fall with 25 students. And with 25 students, you can do all kinds of things that you can't do with 100 students that Adam is going to have. And so those 25 students and I wrote the book. So they figured out the mathematics that was going on. They learned LaTeX. They put a book together. And then last spring, when I was teaching it with a bigger group, I cleaned up the book a bit. This fall, I was not teaching it, but now the book is in reasonable shape. So as you read it, those of you who will go into it, just recognize that, it was put together with people like you. It just gives you the mathematical foundations of much of what we're doing here to the extent that it's possible with with a one-semester calculus background. And for technicalities, Data 8 plus Stat 88 satisfies entry requirements to Econ and Haas. Do you have any questions about any of the connectors or any of the other classes that are going to be offered in the spring before we get back into the swing of things? Yes? That is correct. So the Data 8 website lists this semester's connectors. However, if you go to data.berkeley.edu, simply data.berkeley.edu, the big banner says 2017 classes, spring 2017 classes, they're all there. More questions? Yes?
I saw on the website that to satisfy the econ requirement for HOS, you have to take Data 8 and Stat 88.
Yes. You did see that because that's what it says. So this, what is being pointed out is that the econ website, I believe, says that in order to satisfy the econ requirement, you have to take Data 8 and Stat 88 concurrently. They put that in because they didn't realize that you could not take them concurrently. So it's just fine to take them separately. I've had many students do it separately and satisfy both econ and HOS requirements. Right? We'll talk to econ about that. It's on the econ website that you saw this, right? Okay, I'll look, but it doesn't have to be concurrent. You do have to take both courses for letter grade. More questions? Yes. Can you take Stat 140 after Stat 134? No, you cannot. You can only take one of those for credit. Those are upper division probability courses. 140 requires Data 8, 134 doesn't. More questions? What are the differences between Stat 133 and Stat 28? They're quite different, actually. So Stat 28 assumes Data 8, and you see at this point we're sort of hitting the boundaries of what we can do just with a single variable. So, for example, when you're trying to estimate y based on 1x, you kind of know how to do that, but typically you'll want to use multiple x's. And so multidimensional inference involving multidimensional data, that's going to be their focus. They're not going to be very mathy. They're going to teach you how to use R, but their focus is data analysis, inference. 133 does no inference. 133 is an introduction to using R quite seriously to do, essentially descriptive statistics a little more powerfully than we've done here. 133 satisfies a stat requirement. Stat 28, to my knowledge, doesn't satisfy any requirements at the moment. More questions? Okay, back to the present. All right, so very early on in this class, I think lecture 2, you heard about John Snow and cholera and how he did a version of a randomized controlled experiment. His was a natural experiment. His two water companies were, the houses they served were kind of meshed in like that. And one of them had dirty water and one of them had clean water. And he was able to look at the rates of cholera deaths in the two sets of houses and have a pretty good sense that, there was something in the water. And we talked at that stage about how if you just have an observational study, you've got a treatment group and a control group, that's great. If you see a difference, you can't necessarily ascribe the difference to the treatment if there is confounding. Right? So for quite some time, it was believed that coffee drinking caused lung cancer because coffee drinkers got lung cancer at a higher rate than non-coffee drinkers, but we know now that it was the smoking, right? And those coffee drinkers were also smoking, and the confounding variable was smoking. So to get rid of confounding, what do? We randomly assign the individuals to treatment and control, and then before you start the treatment, the two groups are as similar as possible, at least to the extent that chance allows. And then, so there's no systematic confounding. And then you look at the differences, and perhaps if there are differences between the two groups, you can ascribe that difference to the treatment. Right? That's the point of a randomized controlled experiment. And what does this have to do with what we're doing right now? Well, first, I promised you that day, lecture 2, which you have all forgotten, but I remember, that I had promised you that by the end of this class you will know how to analyze a randomized controlled experiment. And so today that's what we're going to do. What this has to do with what we've been doing thus far is, well, you know, my last lecture seems like a long time ago. It just seems to be floating back somewhere in the distant past. But in that distant past, what we were doing was we were comparing sample A with sample B. Yep, and we were trying to see, these two are both random samples, and we were trying to see this one comes from an underlying distribution, this one comes from an underlying distribution. Are those two underlying distributions the same or not? Yep. And so now we have two groups. We have sample A and sample B, and we're going to compare them. Okay? So that's the point of today's lecture. I'm going to say all of this again in some detail, technical stuff. There's a homework that would have been due last Friday, but we were nice and made it due today. We do want you to do some work on current material. So we're going to post the thing that we should have posted on Friday. We didn't last Friday. We didn't because everybody was on break. We'll post a couple of problems that we'd like you to do by Saturday so that you just don't stop doing anything on Tuesday. Right? Just a couple of problems. Now, there is material that's going to be taught this week, but we can't make anything due on RRR week, so what we'll do is we'll post some practice problems on the current material. And you can do them or not. I strongly advise you to do them. And we'll post answers at some stage, but that's just so that you get a little workout on what we're doing currently. And as I told you, we have tried to devise the curriculum so that you're not learning much that is genuinely new at this stage, though on Wednesday there will be one calculation that's new but straightforward. There's a project due tomorrow. There's been a lot of chatter on Piazza, and looks like it's chugging along nicely. Office hours are on the regular schedule. Mondays and Wednesdays, the rooms have moved from BIDS to Echeverry. Any technical administrative questions before we get going yes? What is happening in lab this week? We have devised a review lab. So basically we want you to get away from using the computer and trying to answer the questions with the computer not there as a guide. So there'll be a little bit of work on the material that's done, I think mostly Wednesday, which is a little different. But then we're putting together review material. So we'll be putting together assorted review materials for you. By the way, next week, RR week, you don't just get your classroom. You have to request your classroom. I don't know what they would do in this room if I didn't request it. Maybe they would have a dance party. I am not sure. But I had to request it. So I requested this room at our class time for Monday and Wednesday. What I'm going to do on those 2 days is I will start on Monday by summarizing all the inference that you know how to do. What hypotheses do you know how to test? What methods were used to test those hypotheses? What were the test statistics? Why? And where that is in the textbook. And then I want to go over and summarize all the sort of mathy things that you learned. Histogram heights and areas starting all the way back. What exactly is a percentile, averages, SDs, Chebyshev's inequality, the normal curve, regression line, correlation, There's a bunch of stuff that you can do calculations with pencil and paper, which tend to pop up on exams. And it's actually a good idea to see what math you actually know and what has been left in the ether for, Adam to sort out in Stat 88 and so on. And that will take us through much of Wednesday, I think, or at least half of Wednesday. And after that, I'll answer questions or do something like that. And the GSIs will be holding specific sessions devoted to this topic or that topic. So there'll be plenty of review. And we'll be posting questions for you to do. So no shortage. They even have, for people who want to come in and go through the experience of taking an exam, they're even going to have an hour session where you can come in and pretend that you're taking an exam. Now, that might work for some of you. When I was a student, I minimized the experience of taking an exam. However, if you on exam days you're not at your best, you might want to do that. So there's going to be all kinds of stuff going on. I don't think there will be any shortage of review. All right, now I actually do need to get to work. One or two more questions. All right. So this is what we were doing last time. I just talked about this. You have two random samples, sample A and sample B, and are they drawn from the same underlying distribution? The method is gloriously named A/B testing. And the way we did it was by permutations. So we said the null hypothesis is that the two underlying distributions are the same. If that were the case, then these two samples that you've got are really not different from each other. You could throw them all in one big pot, permute, and pick out two other sets, and those should look exactly the same as your samples. And so what we did was called a permutation test. We threw all of the two samples into one big combined sample, shuffled, and then split, and then computed our statistic, and then did that over and over again, and compared with what we had observed. So that was to just say, do these two things come from the same underlying distribution or not? And that's fine. That's a question that is asked quite a lot, and it's important to know how to answer it. But in the end, if you have a difference, what you want is some idea of the size of the difference. And so if you have your two samples, you've got sample A and sample B, and your distributions are numerical, which most of our samples are, then you might simply be interested in looking at, what's the difference between the two means, or what's the difference between the two medians. And so there are no hypotheses to make. You've just got these two samples. They have a mean here and a mean there, and you've got one difference. You know that the sample could have come out differently. So to generate your new samples, you just bootstrap your sample. Remember, no hypotheses. You're just computing a replica of your sample, and then you get two different means, and you compare those, and then you do that over and over again, and you have an estimate for an interval estimate for the difference between the means. And that is more or less what we did last time. And so what we're going to do today is to use these methods to see if we can go towards establishing causality. And our two samples are the control and treatment groups, as I just said a bit earlier. And the idea is if those are selected at random, then you can actually make causal conclusions. Not just association, but causation, because you have eliminated systematic bias, systematic confounding. And so if you see a difference between the two groups, then there are two possible reasons for that difference. One simply being chance, which we'll examine in just a moment, and the other being the treatment. But what has happened here is that the third one is missing, which is it could be due to some third thing, like a confounding factor. That's gone because of the randomization. So it's chance versus treatment is what you're looking at. And what we're going to do is see in a particular setting, we're going to analyze a randomized controlled experiment. And actually what I'm going to do is I am going to show you the paper. I'll read that out to you. It's a bit small. All right, so this is PubMed. This is the National Library of Medicine, National Institutes of Health repository of all papers in medical journals. And this one is about a treatment for chronic lower back pain, known as LBP. And what they're using is interesting. They're using botulinum toxin. What's a toxin? What's a toxin? Sorry? Bad stuff. Toxic, poisonous. Botulinum toxin. When you hear botulinum, where does your head go? Botulism. Where does your head go? Plants, okay, fine. So Botox. Yes? That medication is actually a poison. And what it does is it calms down muscle spasms, right? And so ladies who have frown lines can have Botox injected up here, and then the muscles that are contracting like that just loosen out beautifully. And then you have this lovely, smooth forehead, but you can't frown ever again. So, which may be fine for the people around you, but that is a side effect. So this thing is a toxin, and I wanted to show you a phrase in Wikipedia that caught my eye. Botulinum is the most acutely lethal toxin known. All right? This is, as you said, bad stuff. All right? So it's bad stuff. So what they're doing is saying, okay, bad stuff is all very well, but bad stuff used in small doses can actually do good things. And so what they did was they had 31 patients in their study, and all of these suffered from chronic lower back pain, very cleanly done. They randomized the 31, 16 to treatment, and the treatment was just a saline. And the control was just a saline, and the treatment was this thing. It was, as you can see, a randomized double-blind. Can somebody remind me what double-blind means? Randomized, you know what it means. What's double-blind? What's blind? Yes. Exactly. It means, I'm trying to say it as well as was just said by your student here, which is the people giving the treatment don't know which treatment they're giving, and the people receiving the treatment don't know which treatment they're receiving. So both are blind. That, doctors are human too, and they are subject to biases, so if they know that somebody is receiving the treatment, they may be more inclined to say they're getting better. So the medical professional not knowing what is being given is quite important for reducing bias. And of course, the person not knowing what they're given is also important. So this is a randomized, double-blind study. It's about as clean as you can get. And we are going to focus, so they gave this medication and they measured the outcome of the patients 3 weeks after the treatment and 8 weeks after the treatment. And we are just going to look at one of those periods. We're going to look at the 8-week period, and I want you to look at this sentence. At 8 weeks, 9 of the 15 in the toxin group and 2 of the 16 in the saline group had relief. P equals 0.009. Right? So assume that that is correct. It is correct. We are going to check it by our own computation in just a moment. P equals 0.009. What is your conclusion? P, big P or small p? That's a small p, right? So essentially what you're concluding is the alternative. Yes? And what's your gut feeling? What's the alternative? That the treatment does something. Yes? Now, we haven't formalized any of this, but you know by just reading that sentence what the conclusion is going to be. And also you know that because it's a randomized, controlled, double-blind, beautifully done experiment, you have some justification for concluding causality. Okay? So what we're going to do today is simply first figure out exactly what due to chance would mean in this case, and then run the test and see that we agree with this answer. And then see whether we can actually estimate the effect size. How big is the difference between the two groups? And then I want to do, maybe if we have time left over, a little bit of coding just to make sure that we're all clear about that. So before I move to the actual data, are there questions about the setup? There's a statement in there, no patient had any side effects. That is also a good aspect of the study. One of the things that you should be aware of is randomized controlled experiments are, the be-all and end-all of, scientific establishment of causality in the medical world. But when you have a group getting the treatment and a group getting the control, you haven't told them which is which. You haven't told the doctors which is which. But if the treatment has side effects and the control doesn't, then people become aware of what group they're in. Right? And this leads to problems with what is called adherence, continuing with the study. Right? So a person in the control group might decide, oh, I'm not getting the treatment, I won't take it anymore. I won't take the control. But then you've lost your comparison, right? Because there's a self-selected group now walking out of your control group. So this having no side effects is actually quite important to keep your study clean. All right, so data. So the table I'm calling BTA, and that's for botulinum toxin A. And the results, there are two groups. There are 16 in the control group, 2 of which were 1s, and there are 15 in the treatment group, 9 of which were 1s. So this is not a very dramatic data table, but those are the data. And so now my question for you is, if I wanted to find out the 2%, right? What percent of the treatment group recovered and what percent of the control group recovered? What is the Python method that you would suggest that I use? Talk to your neighbor, please, and sort that out. So there are 1s and 0s. I'm trying to figure out the percent recovered in each group. And preferably, I'd like to do it with a short amount of code, if possible. So anybody got a suggestion? Anybody? Yeah. Use pivot, okay? I can use pivot. Shall we use pivot? We'll use pivot. So and like that. Go. Okay. Right. So what you've done is important. What you've done is you have said the results are group 0 and group 1. And in the control group, there were 16, 2 of which were 1s and 14 were 0. So what you've got is the distribution of 1s and 0s in the two groups, right? So you can now convert them to proportions. Yep? It's a fine thing to do. And the other suggestion here was group, right? So again, you have people in two groups, and pivot and group are the two methods that you have for collating results of the two groups without having to do a calculation yourself. And so I'll run group, and what I want in the end is the proportion in each group, yes, of the different categories. And please remember that proportions are means, right, of 0s and 1s. And so if I run group with mean, so run group, this is the label of the categories, and ask for a calculation of the mean, then I have the two proportions right off the bat. So these are two methods that you can use. Yes? I just want you to please connect this 0.125 with what's going on here. What's the 0.125? It is the proportion of 1s in the control group. Yes? And so here, where do I find those people? I find them here. Yep. And it's what proportion? What out of what? 2 out of the 16. 1/8, 12.5. Right? And this 60% is 9 out of these 15. All clear? Okay, the reason I'm spending so much time on this is everything that we've done with 2 samples has this feature. In the end, you have samples labeled by the 2 groups, or classes, or whatever you're calling them. And you have to extract the distributions or the means of the 2 groups. Pivot gives you the distributions. Mean gives you the means. Okay? And so that's really the only difference that we have used, that we've had in calculating test statistics. We've used means when we just have zeros and ones, but if you have categorical data with red, yellow, blue, green, you would use pivot to get the 2 entire distributions, and then you would use total variation distance to compare. But these 2, when you're comparing 2 samples, in your head, you should have, I'm going to either use pivot or I'm going to use group. Okay. And so I'm just going to stick to this because I can see the 2 means. In the control group, 12.5 people had, 5% of the people had a good outcome. In the treatment group, 60% of the people had a good outcome. I am cheering for the treatment right here, right? I'm going, yes, great. But along comes my class, which I have now taught over a period of weeks to be the dampener on everybody's parade. And they will come and they will look at me and say, but it could have been due to chance. Right? And our job now is to examine exactly what is due to chance. How many people are involved in this game here? 31. Exactly 31 people. They were not chosen at random from anywhere. There were 31. By the way, when you sign up to participate in a study, you are not chosen at random and then given a treatment or a control. You have to agree to be in the study. Right? The medical profession has a responsibility to let you know what you're letting yourself in for. So they have to describe that you might get the control, that you might get the treatment. They have to describe the 2 things. They have to describe what might happen, et cetera, et cetera, et cetera. There is a thick set of forms that you have to initial every single page. And after that, you have agreed to participate in the study, and then those people who have agreed get randomized into the 2 groups. So whatever conclusion you make, you are making for those people in your study, and then you have a little bit of work to do to explain why those people are representative of anything. So we'll talk about that in the end. But right now, we're just talking about these 31 people. Right? And the chance is in which one of these got assigned to treatment and which one got assigned to control. And the issue that comes up is this. Supposing you had no treatment whatsoever, right? And you have these 31 people, and poor 31 people are all suffering from chronic lower back pain. Then at the end of 8 weeks, you've done nothing. No saline, no treatment. At the end of 8 weeks, some people are just maybe going to be better. Some people are more, their constitutions are such that they recover. So in other words, there are some people in the 31 who are inherently more able to recover from back pain than others. And if more of them got assigned to the treatment group just by chance, then the treatment results are great, but you don't know whether that's the treatment or whether that's those, what should I say, Teflon people being assigned to the treatment group more than to the control group. And that's the issue. And so this is the thing that needs to be formalized. This is where randomness comes in. It's in who gets assigned to which group. And so what we're going to do is we're going to take a little while to formalize this in terms of null hypotheses. And what helps in such a situation is to imagine each of these 31 patients, before the randomization has happened, they're holding up a ticket, and on one side is their outcome if they were to be assigned to the treatment group, and on the other side is their outcome if they were to be assigned to control. Now, what are these outcomes? These are hypothetical, potential outcomes. And so for the first time, well, actually, this is one example of a model where you are talking about imagined quantities. The regression model was another such. Remember, there was an imagined line, and there were imagined errors. And if the data fit that model, then you could do some inference. So also here, you have imagined treatment scores and imagined control scores. Please look at that. Let me just give you the standard language, is that these are called potential scores. Could you please talk to your neighbor and decide how many potential treatment scores are there and how many potential control scores are there? All the information you need to answer that question is on the slide. You have 31 people in a line. Each one is holding up this ticket, and there are these two potential scores. How many potential treatment scores? Two choices. There were 15 in the treatment group, right? So how many potential treatment scores are there? I'll give you two choices, 15 or 31. This is why we're going through the model very carefully. No randomization has happened yet, right? You have 31 patients, and you are trying to think, if this patient, you're trying to see inherently how likely they are to get better, and you're trying to see if this patient were assigned to the treatment group, what would their outcome be, as opposed to if this same patient were assigned to the control group, what would their outcome be? So these are the potential outcomes. You can't see any of them, right? You can't even measure all of them, but how many are there? In the treatment side, how many are there? How many people are holding up these tickets? 31 people are holding up these tickets, yes? Each ticket has how many sides? 2 sides. How many left-hand sides are there? Okay, let's try pretending we're in second grade. There are 31 people. Each one is holding up a ticket. The ticket has 2 sides. How many left-hand sides are there? 31 left-hand sides. How many right-hand sides are there? 31 right-hand sides. Just because you can't see them doesn't mean they're not there. Right? Notice, these are potential scores. Everybody, before the start of the study, has a potential treatment score and a potential control score. Otherwise, they wouldn't be in the study, right? Because they are going to get assigned to one of those two groups. Okay. So there are 31 of these underlying treatment scores and underlying control scores, and the reason it took so long for us to come up with this was what? Was because we can't see them all. Right? And so what you were worrying about is, well, wait a minute, there's only 15 in the control group. Yeah, fine. Everybody's got a potential score on both sides, and here's what you get to see. For 16 of the people, your control group, somebody comes and puts a big fat hand over the left-hand side, and you only get to see the right. And for the remaining people, the hand goes on the right-hand side, and you only get to see the left. And those initial 16 are a random sample of the 31, which means that the remaining 15 are also a random sample of the 31. It's what's left over. Yes? So what you're seeing is a random sample of the right-hand sides and the complementary random sample of the left-hand sides. And from these, so you're seeing 16 right-hand sides and 15 left-hand sides. And from these, you have to make an inference about what? About 31 right-hand sides and 31 left-hand sides. That's the game here. Everybody has 2 potential scores, but for each person, you just get to see 1. So you have randomly sampled some left-hand side scores and some right-hand side scores. And I'm going to shut up, and I'm going to take questions on this. Yes? How do you decide the potential outcome for each person? You mean, how do you know what number it is? Yes. Okay, so this page back here, these two outcomes are imaginary outcomes. So in our case, it's fixed or not fixed, yes, at 8 weeks. So this is a 1 or a 0, and this is a 1 or a 0, and for neither of them do you know which one it is. No, I'm not doing any randomization here. I haven't done any randomization. I just have the 31 people who have signed the paper saying, I'm going to participate in the study. In my mind, for each of those 31 people, there is a number which I'm never going to see, which is if they get assigned to the treatment group, will they get better or not? And if they get assigned to the control group, will they get better or not? Right? So I'm writing those potential outcome if assigned, but I will never know them. You will never know them. Taiki, the goddess of chance, maybe she knows them, right? But these things are hypotheticals. You had a question. Yeah, so potential outcome, first it's a hypothetical, it's a math quantity, but where from the point of view of the study, what does it involve? It involves exactly the two things that you have mentioned, which is their inherent, their particular characteristics combined with the treatment or the control. All right? And the difficult thing about this is why it causes so much unease, is you're imagining these two potential outcomes. You know that there will be two potential outcomes. So you're just labeling them, potential A, potential B. You have no idea what they are. Yep? So the thing that I have written, potential outcome if assigned to control group, that is what's on the ticket. There's no number. The number would be a 1 or a 0 if I knew what it was, but I don't. Okay, more questions. These are good questions. You can see I've taken an entire lecture to talk about causality, and the whole lecture is dedicated to the model because actually running the test, we've already done that. Okay, more questions. All right, so I have a random sample of 16 people in the control group, and I have a random sample of 15 people in the treatment group. The question, the null hypothesis, is about the distributions of how many people? 16, 15. But the hypotheses are about the distributions of how many people? 31 here and 31 here. Right? 31 potential treatment scores compared to 31 potential control scores, of which I only see part of these and part of these. And I'm still seeing frowns. I will be very patient. If you're frowning, you have a question. Ask me what you're frowning about. A score represents the person's outcome. So if I were measuring the person's weight, it would be the weight as their response to the treatment. In our case, in our particular study, the score is a 1 or a 0. Recovered at 8 weeks, did not recover at 8 weeks. Right? It's whatever outcome you have decided to measure. Right? So it may be blood pressure or it may be something like that. Yes? So the question is, why do I need to do this for the null hypothesis, right? The point is that for my 16 and 15, right, I know they're different. Yes? The issue is that those 16 and 15 could have been a different set of people, right? So where could they have come from? Well, they could have come from the entire 31. All 16 could have come from the entire 31, and all 15 could have come from the entire 31, right? So I have to think about the entire 31, right? So when you think of my 16 control scores, they are a random sample from some underlying distribution of 31 scores, precisely. These 16 scores are a random sample of the underlying 31 scores, and the 15 scores are a random sample from the underlying 31 treatment scores. Those are the two underlying distributions that you are comparing. More questions? How did I decide to choose 16 and 15? I didn't, they did. Right? So I am possibly by tossing a coin. Right? They split 31 in two, and you can't split it evenly. They did 16 and 15. It doesn't have to be even. right? Your coin may give you a little bit off, but that was their decision, not mine. More questions? Yes. The 31 is not random. The subjects who participate in your study are never picked at random. They agree to participating in your study after a very long, drawn-out process. The randomization is in who gets into treatment and who gets into control. That's a randomized controlled experiment. In a randomized controlled experiment, there is no suggestion that your overall group of patients is a random sample from anywhere. They're absolutely clearly not. The randomization is in who gets assigned to which group. That is why we're going through all this here, because maybe the tougher people by chance all got assigned to the treatment group, in which case you would not know how to make any conclusions. Right? More questions? So that's these two things which you never get to see, and therefore you have to write them out longhand in English. The data, now for these 16 people, I actually know these are controlled. So of these 16 tickets, 2 of these are going to be ones, and the remaining 14 are going to be zeros. Yes? And then in these 15 tickets, 9 of these are going to be ones, and the remaining 6 are going to be zeros. So those are very comforting. Those are numbers that I can see, and that was my BTA table. Right? But the point is it could have been a different 16 and a different 15 in the two groups. So finally, the hypotheses, and by now we have talked through this, and so I'm just going to put them up and shut up for a second and let you read. So you have the underlying distributions are the distributions of the 31 potential A scores and the 31 potential B scores. And notice that this null hypothesis and alternative are exactly the same as the hypotheses we had back for the permutation test. We're just being a little careful and saying how many people there are in the two underlying groups. Yep? And so we actually know how to test these hypotheses. We're going to say that if the null hypothesis were true, then you could throw all of those observed 31 scores into a pot, shuffle them, and call any 16 of them control and any 15 of them treatment, and you should get results that are similar. And so you can shuffle, and then you will group, and you will get the mean of your new group and your mean, the mean of the two new groups, and you can compare. And that is what we had called last time a permutation test for the means. And so I'm just taking you through this because it's been a while since we did this. Name of table, the name of the, so name of table is BTA, the name of the variable is result. That's a one, or is, and then there's the name of the class, we're calling that group, right? It's control group or treatment group. And number of repetitions, I'm going to use 10,000 even in class just because we have only 31 total, so you can replicate away. It doesn't take that long. And so then we reduce our table to what we need, and here is the table of means by group and the observed statistic. Okay, observed statistic. Absolute difference between the means. Please tell me why. I'm measuring distance, but why am I measuring distance? What is it that tells me to put that absolute value or not? The alternative hypothesis. That is what tells you whether you're going to put that absolute value. And the alternative hypothesis just said the two distributions are different. That's all it said. Right? And so you stick the absolute value on that. You're just looking at how big, and that's it. And so you shuffle your table, and then you select, and you've got a new table, and then you run the thing again and again, and I went ahead. There's your permutation test. On the horizontal axis, what do you have? You have the test statistic, which is mean of sample A, mean of sample B, minus absolute value. Yes, so that's why they're all positive. Okay? The actual one that we got is, where's the 47.5? 47.5 is 60% minus 12.5%. That's 47.5%. That is what was observed, right? In absolute value, I mean, you can do, if you do it in the other order, you'll get minus 47.5%, but we're taking the absolute value. And so that's here, yes? And therefore, what's your p-value is all the stuff out here, because under the alternative distances are big. What's the p-value that we got?